Good afternoon, BSc second year students. Now we discuss about the enzymes. And last period I have given information about the enzymes. What is meant by the enzymes? Enzymes are the biocatalyst of the living organism in the cell. In the cell of living organisms, that is the simply describe the enzymes. And all enzymes are the proteins, but all proteins are not enzymes. Enzymes is very important characteristics in our life or all the organisms. All enzymes are the present inside the plant cells and animal cells. Yes, and these based the characteristics, nature and nomenclature of the enzymes. And first period I have described in detail about the its nature and nomenclature of the enzymes. Now we discuss the classification of enzymes. In 1961, the enzymes are classified. Enzymes Commission of, that is the IUB, International Union Biochemistry according to the its classification, and enzymes are classified into major 6 classes as follows. And which is the major 6 classes of the enzymes? Now we discuss one by one. Oxidoreductase is the first. Oxidoreductase, first classification. Second class, which method is the transferase. Third one, hydrolysis. Fourth one, lysis, lysis. And fifth, isomerase. And sixth number, ligases, ligases. Student, all of you know that is the I have given information about the last suffix, which may be that is the for examples, oxidoreductases, hydrolysis, transferase. What is the definition of hydrolysis? Hydrolysis, HYDROLYSIS, hydrolysis. But here in the classification, hydrolysis, LASES is the suffix, that is synthesis. Yes, synthesis is nothing but the formation of any other components. I have given that is the in detail, that is the nomenclature. Yes, and that is the base of these nomenclature in the classification. All the classes, last word ASES. Yes, up to the oxidoreductase to lysis. Yes, ASES. Student, you know that is the first we discuss about the oxidoreductase. What do you mean by the oxidoreductase? In this word, two same words related the oxidoreductase. Oxidation and reduction. All of you know that is the, when you remember the simple definition of the oxidation and reduction, or you studied in the 12th science in organic chemistry. What is meant by oxidation or what is meant by the reduction? Yes, the simple definition. It is a simple definition of the oxidation and reduction, and that is oxidation and reduction is related to the enzymes. Yes, oxidoreductase are the enzymes which involve the biological conditions, oxidation and reduction. What is meant by number one? We know that is the oxidation. Oxidation means addition of oxygen or a removal of hydrogen. That is known as the simple definition of the oxidation. Yes. addition of oxygen or removal of hydrogen, that is known as the oxidation. What is meant by the reduction? What is meant by the reduction? Reduction is nothing but the means the addition of hydrogen or removal of oxygen. Addition of hydrogen and removal of oxygen, that is the definition. And oxidoreductases, oxidoreductases, oxidoreductases are classified into two subclasses. Sorry, in the following subclasses. Which is the three important subclasses? That is the dehydrogenases, number 2 oxidases, and number 3 oxygenases. Yes, means oxidoreductases are classified as a subclass as follows. Mind well, number 1 dehydrogenases, number 2 oxidoreductases, and number 3 oxygenases. What is meant by the dehydrogenases? Yes, that is the simple oxidoreductase, the relation, the oxidation and the reduction. Yes, dehydrogenases are the enzymes removal of hydrogen from one substrate to the other substrate. Dehydrogenases are the enzymes which removal of hydrogen from one substrate to another substrate or second substrate, that is known as the dehydrogenases. Yes, removal of hydrogen from one substrate to second substrate. Yes, simple definition. When you describe the simple example of the dehydrogenase, AH2 plus B, in presence of dehydrogenases. Yes, AH2 first and second B. At second to one substrate to another substrate, how to change? BH2 plus A, that is a one substrate to another substrate, that is a change in presence of dehydrogenases. That is a alcohol dehydrogenases, that is the name of the alcohol dehydrogenases. Second one or second type of the oxidoreductases. Oxidoreductases are the enzymes which catalyze the removal of hydrogen, from one substrate and passes directly to the oxygen. That is oxidases. Yes, oxidases are the enzymes which catalyze the removal of hydrogen from a substrate and passes it directly to the oxygen. Yes, what is the simple examples which are related to the oxidases? AH2 plus O2 in presence of oxidase. Yes, AH2 plus O2, and that is the oxidases. In presence of oxidases, AH2 directly to oxygen. Yes, that is simple definition with the suitable examples. Cytochrome oxidase enzyme. Yes, and third one oxygenases, oxygenesis, oxygenases. What is meant by the oxygenases? Yes, oxygens are the enzyme catalyze the incorporation of the oxygen directly to the substrate. directly to the substrate. That is known as the oxygenases. Oxygenases are the enzymes, first mind well, which catalyze the incorporation of the oxygen directly to the substrate. That is known as the oxygenases. That is known as the oxygenases. These types, what is meant by the oxidoreductase? The simple definition of the oxidation, simple definition and its reactions of the reduction and its sub three classes we discuss. And second one is that is the transferases. What is the spelling of transfer? Transferases, Transferases, transferases. What is a simple definition? You know that is the transfer. Transfer is nothing but one place to another place. Transfer from one place to another place, one form to another form. That is known as transfer. Or one transfer from one group to another group. These types related to the its definition. Yes, you know that is the these are the enzymes. Transferases are the enzymes transfer group from one substrate to another substrate. That is known as transferases. Yes, enzymes transfer from one substrate to another substrate. Simply, it is known as transferases. What is the reactions? Simple reaction is represented as the follows, which is the AX plus B. AX plus B. Mind well, AX plus B transferases. AX related, but here A plus BX. BX means X is transferred to the B. X is transferred to B. These types. That is known as transferases. Yes, and simply that is the hydrolysis. That is the hydrolysis. You know that is the hydrolysis and hydrolysis. Hydrolysis means addition of water molecules. That is known as hydrolysis. In the synthesis, that is the lysis, LASES, HYDROLASES, hydrolysis. Hydrolysis are the enzymes. Yes, hydrolysis are the enzymes which catalyze the hydrolysis. That is the direct addition of the water molecules. Direct addition of water molecules across bond, which is cleaved as a hydrolysis and divided into three subclasses. Hydrolysis divided as a three subclasses. Which is the three subclasses? That is the proteases, esterases and carbohydrates. Yes, hydrolysis are divided three subclasses as follows. Number 1, proteases. Number 2, esterases. And number 3, carbohydrates. What is meant by the proteases? Proteases are the enzymes. It is a common word. Proteases are the enzymes that attack to peptide bond. Firstly, I have given in the definitions. Mind well. The first I have given the definition in the all enzymes are proteins. All enzymes of the proteins. That is the proteases. Yes, these are the enzymes attack to the peptide bonds on a protein and peptidases. And peptidases are subdivided into two types, that is peptidases and proteodases. Peptidases and proteodases. What is meant by peptidases? Peptidases are the enzymes. These are the peptidases are the enzymes which act on peptide bond. Which act as a peptide bond adjust to the free amino or carboxylic group. Amino or carboxylic group, that is known as the, or it is also known as the exopeptidases. It is also known as the exopeptidases. And second one, which may be that is the proteinases. Proteinases are the enzymes. Proteinases are the enzymes that attack interior peptide bond of protein. Interior peptide bond of the proteins, that is known as the endopeptidases. Endopeptidases. First I have that is the exopeptidases, and second one that is the endopeptidases. Yes. And third one, esterases. Esterases are the enzyme catalyze hydrolysis of ester linkage. Hydrolysis of ester linkage, that is known as esterases. And these esterases, it is the best example of the liver. Esterases, lipid nucleases, these are the simple examples. And third types, subclass, yes, which is the hydrolysis, that is the carbohydrates. That is also known as carbohydrolysis. These are the enzyme hydrolyze the glucosidic linkage of simple glucosides of the oligosaccharides and polysaccharides. I have given the in first unit in biomolecules, that is the carbohydrates. Yes, and the classification of carbohydrates you remember. Yes, monosaccharides, disaccharides, that is oligosaccharides and polysaccharides. These three types. Remember carbohydrates, and that is the basically. I have given also at that time the enzymes contain protein contain amino acids contain carbohydrates, and yes, and when you detail study about the classification at that time, that words which may be that is the glucosidic, you remember the name, and oligosaccharides and polysaccharides. Basically these are the subclasses of the enzymes, where in the hydrolysis, and last one, that is the carbohydrolysis. Yes, first related to the proteins, second related to the hydrolysis, and third one related carbohydrolysis is linkage to glucosides. Best examples, simple glucosides, that is oligosaccharides and polysaccharides. Yes, and next one, which is the lyses, lyses, lyses. Yes, what is meant by the lyses? Lyses are the enzymes which catalyze either removal of group of atom from their substrate, leaving the double bond, or addition of double bond without hydrolysis. That is oxidation and reduction. That is the oxidation and reduction. Next example, that is aldolysis and fumarosis. These are the examples of the lyses. Lyses are the, remember again, lyses are the enzymes which catalyze the removal of a group of atom from their substrate, leaving double bond, or addition of group to double bond without hydrolysis. That is the oxidation or reduction. Best example, that is the aldolysis or fumarase. fumarase, aldolase, ALDOLASE. Best two examples of the lysis. Yes. And next one, that is the isomerase. Isomerase, ISOMERASES, isomerase. It is the second name of the molasses, MULASES. Isomerases are the enzyme catalytes. Interconversion of compound of one isomers. These are the enzyme catalytes, intermolecules, or rearrangement of the phosphohexose isomers of catalyze the following conversions. Glucose 6-phosphate in presence of the phosphohexose or isomers, that is fructose 6-phosphate. The best examples, students, mind well, I have given the unit first in carbohydrates. Yes, unit first in carbohydrates. That is the what is meant by the isomers. Yes, remember, same molecular formula but different structural formulae. Same molecular formula, the best example C6H12O6. Both the name glucose and fructose. Yes, only glucose presents the hydroxyl group, aldehyde group, and fructose contain a ketonic group. Yes, aldehyde group means ACHO and ketonic group C double bond O. Same molecular form, that is isomers. Iso means same, mers. Yes, that is the same, means glucose 6-phosphate to fructose 6-phosphate is the best examples in presence of the isomers conversion. Glucose 6-phosphate to convert fructose 6-phosphate. Yes, simple. And last one classification, that is the ligases. It is also known as synthetases. Ligases are synthetases. These are the simple one words, might well simple, and that is these are the enzyme catalysts or enzyme synthesis of reactions of two molecules with the breakdown of the pyrophosphate bond ATP to ADP. ATP means adenine triphosphate to adenine diphosphate. Triphosphate to diphosphates. Ligases are the enzyme synthesis reactions by joining two molecules coupled with the breakdown of the pyrophosphate bond ATP to ADP. In common ligases, that is the DNA ligases. Yes, DNA ligases, RNA synthetase and glutamates. These are the simple classifications of the enzymes. Simple classification of enzymes, and enzymes are classified into 6 classes. I have detailed described in the first classification to the last one, that is ligases. Yes, if you read, you listen carefully, and you read the notes, at that time you will know that is the classification. It is very important characteristics of the enzymes. The question may be asked in the exams that what is meant by the enzymes, and describe the classification of enzymes. Yes, at that time you know, you write carefully. If you have any difficult to ask me, and even give me feedback. Yes, now stop here. Good evening. Let it go.